The overall goal of this procedure is to study the chemosensory physiology of individual PEG Sensilla on a scorpion pecten. This is accomplished by first immobilizing an anesthetized scorpion, ventral side up, on a glass slide using modeling clay. To set up requires submerging the pectins in a custom-made oil-filled chamber and making electrodes filled with a chemical stimulant. Chemosensory responses are recorded at individual sensilla on the pecten, which ultimately produce similar response profiles to aqueous chemostimulants.
We used to record from the basis of sensilla while blowing volatile stimulants across the pores. The main advantage of the tip recording technique is that it allows the stimulation to be isolated to one PEG sensillum without affecting the activity of its neighbors.
This method can help us answer key questions in the field of chemosensory biology. For example, why do animals use so many repeated sensilla in chemical detection and perception?
Scorpions might not seem like the safest animals to study. However, not all species are dangerous or difficult to deal with. For example, the desert grassland scorpions are only 4 to 5 centimeters long, are easy to maintain in the lab, and rarely sting if handled properly. If stung, the venom artificially triggers your sensory neurons, some of which transmit pain information. The sensation usually subsides in about 15 minutes. Scorpions are hardy subjects and can endure multiple days of experimentation, often being returned to their habitations when the experiment is complete. To immobilize the scorpion, place it in a pre-chilled glass container. Then place the jar containing the scorpion into a freezing environment for a minimum of